Okay, we're going to pick up where we just left off, and we're going to be talking about synapses just for a short while. A synapse is a site where an impulse is transmitted from one cell to the other cell. So here's like an impulse from this side of this one cell to the next cell, this cell to the next cell. We have two types of synapses, a chemical synapse and an electrical synapse. Electrical synapses are over here. Is this over here. This is where two excitable cells communicate with each other by a direct passage of a current from one cell to the other. When cells communicate this way, they do so via gap junctions. This is a gap junction channel right here. We see the chemical synapses do not have gap junctions. Only electrical cells and synapses have these called gap junctions. Gap junctions are plaque-like structures that gives us a point of transmission that is relatively resistance-free. So over here, we're going to explain the difference to a chemical synapse. This chemical needs to be released, move through the substance to the other side on the postsynaptic side. Here, this signal can go back and forth with great ease with no resistance. It doesn't need to go through any sort of substance in the middle over here in the cleft. So to reiterate, gap junctions are these plaque-like structures right here. That gives us a point of transmission which is pretty much relatively resistance-free. So this signal can go from one cell to the other extremely rapidly and very efficiently. Chemical synapses are over here, and this is what we're going to be talking about. This is what happens at our motor end plate. This is how our nervous system and our muscle cells communicate with each other. We use chemical synapses, not electrical synapses. Electrical synapses are not at the neuromuscular junction. Chemical synapses are. So these are suited for complex activities because you have so much more potential for regulation of the transmission of that signal. Over here with the electrical synapse, you really don't have much control. It's going to go through those gap junctions and so forth. But if we needed to alter something on the postmembrane side, this gives us a tiny bit of time to do so. So we have a lot more control at this synapse than we do at the electrical synapse. They are more appropriate to use as activities become more complex. These give you more control of what happens on that postsynaptic side. Here we need a chemical neurotransmitter to initiate an action potential. This initiates the action potential from the presynaptic side to the postsynaptic side. There is no physical contact at a synapse that is chemical in nature. Since there is no contact, we need some sort of other way of transmitting this signal. We don't have gap junctions here, like the electrical synapses have. We have a space through which the signal needs to be transmitted. And right here, we have the space through which it needs to be transmitted. This does not have a space. So in order for a chemical synapse to be effective, there must be appropriate receptors on the postsynaptic side. The neurotransmitter being used will vary depending where we are on the body, and that will determine what receptors will be activated on this postsynaptic side. In order for the synapse to be effective, there must be some way of transmitting that signal. So there's a synaptic cleft that's located in the chemical synapse, which we have been talking about right here in the middle. This synaptic cleft is filled with an amorphous substance which is rich with carbohydrates. What does amorphous mean? Amorphous meaning that this substance is pretty much free-flowing and will assume the shape of the container in which it resides. This amorphous substance will facilitate this transmission of the signal from the presynaptic side to the postsynaptic side. The amorphous substance will facilitate this transmission because it gives the chemical something through which it can pass. It's not just a free-floating space. There's actually a substance there, and again, that is rich with carbohydrates. The signal actually needs to pass through this substance. So continuing with chemical synapses, all chemical synapses have what's called a synaptic delay. It takes time to initiate all of these activities. So the chemical needs to be released from the presynaptic side, which is right here is our storage vesicles. We're going to get into the steps in a second. But ultimately, we need to release the substance, which When we're talking about a motor end plate, we're going to be talking about acetylcholine. This chemical needs to be released from the presynaptic side, needs to go through this amorphous substance and attach to the receptors on the postsynaptic side in order for an action potential to be propagated. Needs to bind to the appropriate receptors. Over here, we're going to be talking about alpha receptors, and trigger the appropriate postsynaptic side activity and for the signal to be carried. So once we have acetylcholine, we're going to go through the synaptic cleft over here. We're going to attach to alpha receptors, and then that attachment of acetylcholine on this postsynaptic side is going to alter the membrane permeability over here, and we're going to generate an action potential on the postsynaptic side. We're not going to go too detail of how this occurs physiologically, because there are a number of other steps we could go over, but ultimately I want you to know what the basic steps are of how this occurs. So this is not the case for electrical transmission synapses. There is no synaptic delay, because we want that signal of the electrical synapses to be resistance-free and want it to recur with great speed. If you could guess where electrical synapses occur, that would be the heart, right? Because we want the heart, we don't want any synaptic delay or anything that could happen that could alter our cardiac tissue beating, right? We'll get over that. We'll go over that a little bit once we start talking about cardiovascular physiology. However, if a skeletal muscle doesn't fire, it's not the end of the world, or fire all at once. However, if something were to occur at electrical synapse, just like in the heart, we want the heart to beat as one in sync with each other. So it has to be fairly resistance free. So an example of where those electrical synapses would be within the myocardium itself. Okay, so the major differences between different chemical synapses. So chemical synapses from one to the other, not electrical. There's different types of chemical synapses, and here are the differences. 1, how the presynaptic side and postsynaptic cells are structured. 2, what neurotransmitter is going to be used. And 3, the actual structure of the synapse may be different itself. So again, in this class right now, we're really only concerned with this skeletal muscle function between the nervous system. So we're going to be talking about the motor end plate as well as the alpha receptors and acetylcholine that's going to be causing this action potential on the postsynaptic side. Okay, so we're going to be on the presynaptic side, and first we need to start talking about the axon at this neuromuscular junction. Again, this is where the nervous system is meeting the muscle cell itself. So when we talk about the nervous system, we're talking about the motor unit and so forth. And the nerve that we're talking about has a body, so a soma. Here's the cell body, which is the soma, has a bunch of dendrites, and then we have one big axon and axon collaterals. So axon collaterals, so here's the axon, it's coming off of the soma, off of the body, and then we have a number of axon collaterals coming off of it. Each of these collaterals has a terminal branch, which you can see at the end has a little bulb. It's called a synaptic bulb. That's what we care about, and that's where the motor end plate is going to be. A number of these axon terminals impact many more cells than if it were just to have one axon terminal at the end of each axon. So this is actually a benefit to us. One thing that distinguishes an alpha motor neurons between slow twitch and fast twitch fibers is the thickness of the axon itself. The thicker the axon, innervate our type 2 fibers. So those are our big, powerful fibers, right? Our anaerobic fibers. And we want that transmission to be extremely quick, right? So if we're doing a vertical jump or lifting one heavy weight or 1 rep max, we're going to be activating more of those. The thinner axons tend to innervate more type 1 fibers, which are our oxidative fibers and our slow twitch fibers. Right. So let's talk about nodes of Rambier. That's what this sheath is on this axon here. They're on axons that have myelin sheath and allows the signal to hop from one location to the other with great speed. So this myelin sheath that is on the axon allows that neural signal to transmit extremely quickly down the axon, into the axon terminals, into the bulbs. As you get closer to the end of the axon, the myelin sheath disappears. So you can see right here, myelin sheath eventually stops, and that's going to be a big component to this neuromuscular junction with regards to the steps we'll be talking about. Where the myelin sheath is absent, you will find voltage-dependent gates. These voltage-dependent gates are critical to synaptic transmission at the neuromuscular junction. So right here, we're going to zoom in 1 second, but right here where the myelin sheath stops, we're actually going to have voltage-dependent gates, and those are critical to trigger synaptic transmission at this neuromuscular junction. At these bulbs right here, these bulbs are going to be the ones that are connecting at that chemical synapse with the muscle cells themselves. Okay, so talking from here, when we look at this diagram, this is the bulb, the synaptic bulb at the end of that axon terminal. And that's the bulb right here, zoomed in. So if you could get a picture of that, for the most part, we have the soma, the axon coming off, axon collaterals, and then we have this synaptic bulb at the end. And here it is. Here's one synaptic bulb innervating with, or communicating with a skeletal muscle cell right here. The more skeletal muscles you want to innervate, the more terminal bulbs there will be. Makes sense? If we, again, want fine motor control, axons will only innervate a small amount of muscle fibers to give you more control. So just like we said before with the eyes, if I want to move my eyes up and down, or you want to do digital manipulation as you're typing on your keyboard, you want to have fine motor control. So one axon will only innervate a small amount of muscle fibers. Now, in larger muscle fibers, such as the glutes, again, there may be thousands of muscle fibers being innervated by just one axon or one bulb. So here, inside this bulb, so now we're in the synaptic bulb. Inside the bulb, there's vesicles. You see these synaptic vesicles? Filled with a neurotransmitter called acetylcholine. So these little red molecules that look like in these vesicles, that's acetylcholine in there. One synaptic vesicle contains a quantum of acetylcholine. Couldn't tell you how much a quantum is, but it's a lot. So inside also the synaptic bulb, we have something that's called an inactive enzyme. So it's free floating right here. So an inactive enzyme is actually free floating right here. Just all throughout here. And it's just called inactive enzyme. There is no name for this enzyme. The switch that turns on synaptic transmission is conversion of this inactive enzyme to an active form. Okay. So now moving on to the postsynaptic side. So now we're here. This is the motor end plate. We're at the muscle cell right here. This is where the axon bulb is. Here's our cleft with our amorphous substance in between, rich with carbohydrates. And then here we have our muscle cell. This is where the motor end plate is. The motor end plate is not a smooth structure. It has junctional folds in it. You can see how we can kind of go up and down in these junctional folds. The receptor configuration on this side, this postsynaptic side, is going to determine if the signal is going to be accepted or not. There's 4 different types of receptors there. We have alpha receptors, beta receptors, gamma receptors, and sigma receptors. They're all up and down these troughs. So the alpha receptor is duplicated. So we have double the amount of alpha receptors at the motor end plate. That's a good thing because that's what acetylcholine wants to bind to. So once we release all of this acetylcholine into this cleft, acetylcholine is going to attach to the alpha receptors, and that's what's going to give us our transmission. The alpha receptors sit on top of the folds, which is more efficient for us to attach to. On the postsynaptic side, we have cells that are inside negative based upon the relationship between sodium and potassium. Again, so it's going to be the same thing on this side of the cell. Okay, so once we have our signal coming down here, there's going to be a series of events that occur to release acetylcholine into the cleft, attach to the alpha receptors, and then again, we have cells over here that are inside negative based upon our relationship again to sodium and potassium. And we want to alter that relationship again in order for the signal to keep transmitting. All right, in our next video, we're going to talk about the steps of all of that.